you know what DNA fingerprinting is?
Yes, I do. It's a technique used for identification. For example, you see my phone right here, like you can fingerprint.
Do you know what DNA fingerprinting is? DNA fingerprinting is the analysis of DNA from samples of body tissues or fluids, especially when conducted in order to identify individuals.
DNA fingerprint paternity testing. DNA fingerprinting with gel electrophoresis can be used to determine who the parents of an organism are. Just like genes, DNA fragments are inherited from both parents. What is DNA? Stupid facts. If two different people started reciting their individual genetic code at a rate of 1 letter per second, it would take them almost 8.5 minutes before they reached a difference. If unwounded and tied together, the strands of DNA in one cell would stretch almost 6 feet, but would be only 50 trillion of an inch wide. If all DNA in your body was put end to end, it would reach the sun and back over 600 times. 100 trillion times 6 feet divided by 92 million miles.
How does DNA fingerprinting work?
The root from a single strand of hair is enough for researchers to work with. The sample contains white blood cells which are broken up open using detergent, and all the usable DNA is separated from the extracellular material. Next, the restriction enzymes are used to cut the DNA into smaller pieces.
For example, on the right side, I have 5 different images of fingerprints. The first one is, as you can see, is arch, loop, and rule. As you can see, they're all 3 different. Nobody has the same fingerprint in the whole entire world. It's impossible. As you can see on the left side, there's a crime scene. For example, if something happens or a crime scene happens, people can go and they can trace fingerprints and see who is who and where these person is or all that type of stuff. So as you can see, there's a crime scene and they have 3 suspects. The 3 suspects all have different fingerprints, but as you can see, one matches the crime scene, and that is also suspect too. So yeah. As you can see, it's very easy to find fingerprints. All the police have to do is go swatching for fingerprints, and you cannot be duplicated. So that person suspect too had to be caught. As you can see, their fingerprints match the crime scene.
My house was broken into last week. The police can identify the culprit from blood found at the scene. How can they do that? To understand this, we need to state what is meant by DNA fingerprinting and describe some uses of DNA fingerprinting. The burglar smashed a window, leaving blood on the broken glass. Samples of blood, hair or skin can be collected from a crime scene. Forensic scientists investigate the samples in a lab. First, they extract DNA from the cells. They use enzymes to cut the DNA into shorter fragments. The fragments are placed into a well in a gel. They are separated using an electric current. Different sized fragments travel different distances in the gel. The smallest fragments travel the furthest. A pattern of bands is produced. This can be compared with the patterns of DNA from possible suspects. The enzymes cut different people's DNA in different places, producing a different set of fragments. The different sets of DNA fragments will produce different patterns of bands when separated on a gel. Each person has unique DNA.
DNA fingerprinting was developed in 1984 by Alec J. Jeffrey at the University of Leicester while he was studying the gene of myoglobin. He found the myoglobin gene contains many segments that vary in size and composition and have no apparent functions.
Are you riding? Say you never ever leave from beside me. Cause I want you, and I need you, and I'm down for you always, KB. Do you love me? Are you riding?